our Chapter 3 audio lecture. So in Chapter 3, we're going over all the cells. We're going to break this one up into two parts just to make it a little more manageable and make it where you don't have to listen to me talk for too long a period of time. Okay, but the first thing you always want to do when you get to a new chapter is sort of just think about, why is it important that I know this chapter and why is the information so important? And when you're talking about cells, it really comes down to cancer. Okay, so cancer is a group of diseases characterized by uncontrollable and abnormal cell division. So when you want to break down and sort of why do people kind of get cancer and what's sort of happening physiologically with cancer, it really does just come down to this cell behavior. Okay, so approximately 38% of men and women are going to get cancer at some time in their life. And we all have heard about chemotherapy and radiation therapy, but what happens with those is that we're going to be able to cause this death of these cancer cells and allow your normal cells to kind of continue reproducing and kind of doing their job properly. But what you're trying to do is sort of block this rapid cell division of these sort of cancer cells. Okay, so your cells are all supposed to have a certain amount of timeline for how long they're supposed to live for and how long they're supposed to sort of replicate for. And what happens is when they kind of start reproducing and sort of dividing for longer periods of time, they start to kind of make mistakes, and then these cells can kind of take over and make it hard for those organs to kind of function. Okay, so this is why this cellular information, just the basic principles, are going to become so important in our efforts to sort of cure cancer. Okay, one of the other questions I got was, we talked about we have these normal ranges for maintaining homeostasis, and we're trying to sort of stay within these narrow limits. Temperature is a great one for that. Okay, we all know that our normal temperature is going to be this 98.6 degrees. But you end up, if you're too cold or you're too hot, you really just have a few degrees in there before it gets really, really dangerous for your body. Okay, now we all think about 98.6, but everybody's a little bit different. And what you really want to know is kind of what's normal for you. So the normal temperature for a person can range between 97.1 and 99 degrees. And again, it's important, especially now where you're taking your temperature and sort of knowing like, okay, I'm normally 99 and now I'm higher, now I'm kind of low. Okay, so you want to seek medical attention. This is the questions I always get from patients is you want to seek medical attention for adults if your temperature gets over 103. Okay, for 6 to 24-month-olds, if it's over 102 and it lasts for more than a day, this is always a hard one where you call your pediatrician. They're like, oh, my child has a fever of 102. Should I come in or not? And they say, okay, well, wait a day and see if it's 102 kind of tomorrow, which is always a little bit scary, especially for like a newer parent. For 3 to 6 months old, anything over 102, you want to seek medical attention and take them to the doctor. And then for a 3-month-old and under, okay, anything over 100.4. So again, thermometers, especially ones you do at home, are pretty inaccurate. And everybody knows, especially now where we're taking our temperatures all the time, you can really do 3 readings and they can all be a little bit different. But again, you're just varying between 3-4 degrees and it becomes really, really important really quickly. So it's just an amazing thing that your body does is that it can maintain this homeostasis and maintain these optimal temperatures within a very, very small margin. Okay, another question I get all the time is ibuprofen. Why do we take ibuprofen for fevers? Okay, so they're going to limit the production of prostaglandins, and this is one where you just want to kind of go look it up real fast. But these are going to be hormones which are released in response to injury or illness, and they're going to produce inflammation, pain, fever, and they're just part of our healing process. So a lot of times we think of inflammation as a bad thing, but a lot of times you want to remember that is how our body kind of fixes things. But ibuprofens are just going to limit this production of these prostaglandins that create all these kind of symptoms. And then another question I get is aspirin versus Tylenol. Should I take one versus the other? What you want to think about with these is that Tylenol is actually working on your perception of pain. So this is my acetaminophen. Tylenol, acetaminophen, they're actually just working on the way your body processes pain. So they're not actually working on the area that's injured. Aspirins are going to be a little bit more of an anti-inflammatory, and they're going to work on the spot that's actually kind of injured. So if you can tolerate aspirin, because a lot of times this comes down to what can my stomach tolerate normal aspirin or ibuprofen or different kind of things. And if it can't, then we have to move over and we have to start looking at the acetaminophen and the Tylenols. Okay, but everybody has one that kind of works kind of better for them. Okay, so one is perception of pain, and one is actually working on the area that's a little bit injured. Okay, so now let's get into chapter 3, which is our cell. Here's a good picture that you want to make sure you can label, especially for our practical. This one's going to come up for sure, and you just want to be able to label all the things that are on your objective list, but all the things that we're going to kind of talk about today, and then we'll come back to this one a little bit too. So here is our cell theory and our cell principles. There are over 50 to 100 trillion cells in the body. That's an astronomical number. We have 250 different types of cells. Regardless of their differences, all cells are going to have the same basic parts and the same common functions. Human cells are going to have three main parts. They're going to have a plasma membrane, a cytoplasm, and a nucleus. And again, we want to be able to label them, but we want to know what the basic function of all these different little parts are going to be. Okay, so your plasma membrane is the outer boundary. It's going to be selectively permeable. We're going to talk about that a lot in this first part of our audio lecture. Number two is the cytoplasm. This is the intracellular fluid, so the fluid that's on the inside of the cell. And then we're also going to have our nucleus, which is going to be an organelle. Most of us are familiar with the nucleus, but it's going to control all cell activities. Okay, so your plasma membrane is going to separate intracellular fluid within the cell and the extracellular fluid on the outside. So again, it's creating this barrier between interior and exterior. It's a double layer of phospholipids. We're going to see a picture here in a second, which is embedded with proteins. Many proteins float around. They're not anchored in there all the time. They're going to float around in the membrane, and they're going to create this constantly changing pattern, okay? But we end up with this sandwich-like structure or lipid bilayer. Important multiple choice question. Consists of two parallel sheets of phospholipid molecules, and when you hear lipid, you want to think about fats, and these are going to lie tail to tail, okay? And again, this is one of those things where we're looking at the interior of the membrane versus the exterior of the membrane. Their polar heads are going to be bathed in water on either side, and this allows them, again, to just be in contact with the internal environment and the external environment. Okay, so here's a picture of our membrane, and I can see my phospholipids here, and I can see that they have a head and a tail, and I'm orientating these heads outward into the extracellular environment and inward into the intracellular environment, and I can also see that I have a lot of these proteins embedded in there, and we'll get into why that's important in a second here. So the phospholipids have a polar head. It's going to be charged. It's going to be hydrophilic, so it's going to be water-loving, because again, I want to be able to sample and move things around. The nonpolar tails are going to be uncharged. They're going to be hydrophobic, and this is they're water-hating, and this is kind of how I create this barrier, okay, is that I can kind of repel certain things. The polar heads attract water, which is the main constituent of both intra and extracellular fluid. So again, they're attracting that fluid to the surface. It's going to lie on the inner and outer surfaces of the membrane. My nonpolar heads, again, they're going to avoid water. They're going to line up in the center of the membrane. Okay, so my plasma membrane also is going to contain glycolipids, and again, I just want to know that they're in there, and I want to know what their function is, but their main job is that they're going to help me sort of stabilize my membrane. And you also want to realize that there's cholesterol, because we always think of cholesterol as a really bad thing, but it is important to every single cell. It's going to make up 20% of that plasma membrane, and it's going to help me sort of stabilize my membrane, okay, and keep things from moving too easily. So now we get back to our picture, and again, I just want to find all the things that we just had to kind of label, right? The heads, the tails, all the different little kind of channels. I want to find the external environment, find the internal environment. And now we want to move on to a little bit more information about these proteins that are embedded in the membrane. Okay, so membrane proteins within the plasma membrane, they're allowing for communication with the environment, and the main thing they're doing for us is they're going to allow us to transport things across the membrane. Okay, so they're going to make up 50% of the plasma membrane. That's a huge number. Again, some are going to float freely, others are going to be tethered, but again, we're just creating this way that I can move things in and out of the cell. So you get two distinct types of membrane proteins. Again, good multiple choice question. You have integral proteins and peripheral proteins. The integral ones are going to be firmly inserted into the lipid bilayer. Okay, most are transmembrane proteins, which means that they're going to span across the entire membrane and they're going to protrude from both sides. So that's, again, a good multiple choice question is, what's the difference between these two? And, do they span across the whole membrane or do they only go part way? Okay, but because they span across from one side to the other, they're going to be involved in transport. So they're going to help me to form these channels and these pores. They can act as receptor sites. They can act as enzymes that are going to assist in reactions, and we'll come back to some of those in a second here too. Okay, peripheral proteins are a little bit different. They're not embedded. They're going to be attached loosely to the integral proteins. They're easily removed without disrupting the membrane, and they're going to be involved in membrane support, enzyme activity, cell shape, muscle cell contraction. But again, this is just another way that my body can kind of control exactly what everything is doing at all different times. The plasma membrane, we said this before, is going to be selectively permeable. This is going to come up in a bunch of questions here too. So how do we get things into and out of our cells? We're going to talk about passive versus active transport. Passive, you want to break it down to just as simple as possible. It does not require energy. Active is going to require us to use energy. Okay, and a lot of your questions for your final and for your lecture exams are going to come back to, do I know which ones are passive and which ones are active? And, do I know, okay, one requires energy or one does not require energy? So very, very important. Okay, passive transport, you get two main types. You get diffusion, you probably heard about that in high school, and we're also going to get filtration. Okay, so anytime you see diffusion, you want to remember that it's going to be passive, okay, because diffusion is a really, really important one. And this is what we're talking about when we talk about diffusion. It's the tendency of molecules or ions to move from where they are in a higher concentration to an area where they are in a lower concentration. So they're moving from where there's a lot of them to an area where there's less of them. So this is also going to be termed that they're moving down or along their concentration gradient. Okay, and that's another good multiple choice question. Is it diffusion or passive stuff is going to move down this concentration gradient, and again it's moving from a high concentration to a little bit of a lower concentration, and again they're basically just trying to find a little more room. They're trying to balance things out on either side. This is going to happen due to a few things. So with diffusion, we have the constant and random high-speed motion, okay, causes collisions between particles. We're going to look at a picture here in a second. With each collision, the particles change direction, and this is going to cause them to sort of disperse, okay? So the overall effect is to disperse the particles throughout the environment. Greater concentration is going to equal more collisions. So if I have a lot more particles, they're going to bang into each other a lot more. It's going to help me with my faster diffusion. It's also going to be influenced by particle size. So if they're bigger, they're going to run into each other a little bit more. And it's also going to be influenced by temperature. Things are going to occur faster at a higher temperature. Okay, so here is a picture of a closed container. We have a uniform mixture. We're just taking a solution. We're going to drop a dye pellet in. There's nothing restricting the diffusion, right? So the dye pellet's going to diffuse. It's going to spread out until I get this nice sort of uniform mixture. And because there's no barriers, it's going to be termed this free diffusion, okay? Now, our plasma membrane does not work like that, right? It's a barrier to free diffusion. So we've already said that our plasma membranes are selectively or differentially permeable. So they're going to allow some substances to pass, but they're going to exclude others. And this allows us to kind of keep valuable substances in and keep undesirable substances out, or bring things that we need in or move things out that we don't want anymore. Okay, so you got to remember that our plasma membrane, it's not free diffusion, it's going to be selectively permeable. It's only going to allow certain things to move in and out. Okay, so if the plasma membrane becomes severely damaged, as in with a bad cut or with a burn, this becomes a problem because now everything is allowed to sort of get across this little barrier, and then this is where we can get a lot of different infections and sicknesses and things like that. So really, really important to sort of maintain these barriers. Okay, so molecules and ions diffuse through the membrane if they are lipid soluble, so fat soluble, small enough to fit through the membrane channels that we're creating, right? Or they're going to be assisted by a carrier molecule. Okay, so another good multiple choice question is how are, molecules and ions can diffuse through a membrane, and then they'll list these and give you one that's kind of not included in there. So types of diffusion, we're going to have simple and facilitated. And I just want to mention this real quick because a lot of people kind of get this wrong. As soon as we see facilitated, we start thinking that we need energy. And you want to just remember that, I want to be able to kind of list everything and keep them in their proper categories. But facilitated diffusion is still a type of diffusion, and that means it's still going to be passive. Okay, but that's one question that people get wrong in the multiple choice all the time. Okay, so types of diffusion, simple versus facilitated. So simple diffusion, nonpolar and lipid-soluble substances diffuse directly through the lipid bilayer. A good example of this is oxygen and carbon dioxide. And you're going to find that with the passive things, it's usually things that our body needs a lot, okay, or things that we have to move around a lot. And oxygen and carbon dioxide are obviously one of those things. But oxygen concentration is higher in the blood, right, because we've put it into the blood to be able to transport it around to the body. And it's going to be lower in the tissues, okay, because we want to actually put it into the tissues so that they can use it, right? So oxygen is constantly diffusing into our cells kind of from our blood. Carbon dioxide is, again, the opposite, right? It's going to be a byproduct. It's going to be more waste, right? It's going to be higher concentration in the cells because that's what they're going to produce as they use this energy. And so we're going to move it in the opposite direction, right? We're going to move it from the cells into the blood, and then we can transport it back to our lungs, get rid of the carbon dioxide, put more oxygen into there, and then move it back to all of our tissues. Okay, so that's a really, really good example, and then we'll talk about sort of why that's so important in a little bit too. So here is a picture, and again, you want to have that medical dictionary, because again, as I'm going through, I might see something that I'm not sure, and in this one you might see, hey, lipid-soluble solute, and you might think, oh man, I don't really know what a solute is, so this is where you want to go back and kind of look it up real quick, okay? But a solute is a substance that is dissolved in a solution, okay? And this is just showing you the barrier, the extracellular fluid, intracellular fluid, and it's just showing you how things can move from one side to the other. Now we get to facilitated diffusion, again, still passive. Certain molecules are transported passively, even though they are unable to pass through the lipid bilayer. A transported substance can either do one of these two things. It can bind to a carrier protein, okay, or it can move through a protein channel. And we're going to take a closer look at these ones too, but again, it's still a type of diffusion, so still passive. So here is my carrier-mediated facilitated diffusion, and just by the name it kind of sounds like it's going to require energy, so again, can be a little bit tricky. But carriers are transmembrane integral proteins. They're transport molecules too big to pass through membrane channels. There's going to be a binding, okay, so you have a binding of a substrate. It's going to cause an alteration in that protein shape. The transport protein then can envelop and release the substance into or out of the cell. But again, we're still moving down this concentration gradient, what makes it passive, okay? Alright, so here is our little picture of carrier-mediated facilitated diffusion, okay, but still moving down that concentration gradient. A good example of this one is glucose, so another thing that our body needs a lot. So normally a higher concentration in the blood than in the cells. So we eat our food, we break everything down, we move this glucose into our blood. But again, our cells need glucose for energy. They need to be able to sort of create ATP. So we're going to use this rapidly for ATP synthesis and energy. This one's going to be a little more limited, okay, by the number of protein carriers that are available. Okay, so I have to have all these little protein carriers to be able to get this glucose kind of where I need it to be. But another good example. So channel-mediated facilitated diffusion, also transmembrane proteins. Usually they're going to transport ions or water. They're going to be selectively permeable, again, selective due to pore size and charge of the channel. We have two types of these. We're going to have leakage channels. These are going to be always open. Okay, and then we have these gated channels, and these are going to be controlled by different chemicals and electrical signals, and they're going to open only under certain circumstances. Okay, and we'll come back to this, especially when we talk about firing our neurons and how our nervous system kind of works. They can become inhibited, they can become saturated, they can be very specific, but again, just another way that our body can control what's moving in each direction and control what's kind of going on. So here's a picture of channel-mediated facilitated diffusion. Okay. And then now we move to why this is all important. So passive transport by diffusion is vital to cellular homeostasis and balance. And again, this is what it comes down to. Because it's passive and it doesn't require energy, it saves us a tremendous amount of cellular energy. And we just want to think about how much energy will we have to use to sort of move oxygen and water and glucose and all these different things around all the time. Okay, so it would be one of those things where we would need to take in an enormous amount of energy to be able to sort of run all these things if they were not passive. Okay, but I'm transporting oxygen, water, glucose, and various ions by this passive transport. Okay, so now we get to osmosis, another term you probably heard of in high school. It's going to be the diffusion of a solvent, okay, such as water through a selectively permeable membrane or channel. So most of the time when you hear osmosis, you're talking about this movement of water, but we're going to end up with these little terms that again show up on your tests all the time. Okay, and again, we want to be like, okay, diffusion of a solvent. Do I know what a solvent is? Okay, and I want to go, if I don't, I want to go look it up. A solvent is a liquid capable of dissolving another substance, and then the solute we already talked about is the substance that's going to be dissolved in a solvent. Okay, and again, we're talking about this external versus internal fluid. Osmosis occurs whenever the water concentration differs on each side of the membrane. Okay, so again, it comes down to this internal versus external environment. Okay, and here's just another little picture here of osmosis and diffusion of the solvent. And now we get to this important picture here. Okay, so this is a membrane and it's permeable to both solutes and water. So both of them are allowed to move. And in this picture, again, we have this little membrane, but it's freely permeable, means everything can move. Okay, so in this picture, I have more solutes on this side, and I have less on this side, because everything is allowed to move, right? I'm just going to move the solutes until I have this equal concentration on both sides. But again, this is free freely permeable membranes, which is again going to be different from the way our cells are going to work. Okay, so this is how our cells are going to work. So this is where I have a membrane. It's permeable to water because we said water can move easily, but it's impermeable to the solutes. So in this instance, I have this blockage here with this membrane, and I have a lot of solutes on one side, and I have a lot of not as many solutes on the other side. And again, the only thing that I can move to make these concentrations equal is I can move the water. So in this picture over here, you can see that I've moved the water to the other side. So the concentration of solutes in each of these solutions is going to be equal. Okay, but again, it's the water that's going to move because I'm not allowed to move the solutes in this case. And again, that's what's happening kind of in the body. Tonicity is another term, but because many solutes cannot diffuse through the plasma membrane, they can build up on one side or the other. Okay, so any change in solute concentration is going to alter the water concentration on both sides of the membrane because, again, I'm going to move it to make things equal, and it's going to result in a loss or gain of water by the cell. Okay, so tonicity is the ability of a solution. So if I put a cell in a type of solution, is it going to change? Okay, so is the shape or tone of the cell going to change? And again, it's done by altering the cell's internal water volume. I end up with three types of solutions, and again, you probably looked at these in high school. Okay, but I end up with an isotonic, hypertonic, and hypotonic. And again, what you want to remember is you want to kind of know the definition of these types of solutions, but you want to know what happens to the cell. Okay, so isotonic, the solution that we're putting it in has the same concentration of solutes as found in the cell. Because they're the same, okay, they're going to retain their normal shape. You're going to have no net loss or gain of water. Okay? Hypertonic's a little bit different. So in this one, I'm putting it in a solution that has a higher concentration of solutes than seen in the cell. And again, I want to try to balance things out. So what I'm going to do is I'm going to take the water from the cell and I'm going to move it to the outside. So when immersed, cells are going to lose water and they're going to shrink. And then hypotonic, this is a more dilute. The solution has a lower concentration of solutes than in the cell. And what happens with this one is that the cell is going to plump up as the water from the outside is going to kind of rush in. So again, it's all based on what type of solution, which one has more or less concentration of solutes, and then where is that water going to sort of move to. So they give you this nice little diagram here where it's easier to remember. Okay, so isotonic stays the same. Hypertonic, it's going to shrink. And then hypotonic, it's actually going to swell up a little bit, okay, as that water rushes in. But good multiple choice questions, okay? And again, just things that you want to just make sure, terms that you want to make sure you know for especially for your lecture exams. So hypotonic solutions, this is just an example here, but they're going to be used in mild cases of dehydration. And again, this is my sports drinks, okay? We're going to use them to rehydrate tissues. But what they do is they create these so that the solution has a lower concentration of solutes than in the cell. And again, when I expose these cells to this type of solution, what's going to happen is that the water is going to kind of rush into the cell, and then I can rehydrate a little bit. Okay, so just a little bit when you're drinking your Gatorades and all your other stuff, sort of knowing why they work so good. Okay, now we get to active membrane transport, and this is going to be the last part of our part one of our audio lecture. So we looked at all the passive stuff. Now we just want to take a quick look at the active stuff, and then we'll come back for part two and we'll look at sort of what we find in the cell. So active membrane transport, an active process that occurs when a cell uses energy, that's the most important thing, to move solutes across a membrane. And again, they're either too big, they can't be dissolved in the lipid bilayer, and the important thing to remember with active stuff is that they're going to move against the concentration gradient. So remember we said one's moving from high to low, this one's going to move where they're going to kind of move where they're more sort of crowded. So we do this in two ways. We use active transport and we use vesicular transport. Very important to remember that these two are part of active. Okay, so you want to remember vesicular is part of active transport. Okay, so active transport requires a carrier protein, moves solutes uphill, okay, so they're moving against the concentration sort of gradients. It's going to require energy, and the best example of this one is going to be the sodium-potassium pump, and we're going to come back to that one again, especially when we get into our muscle chapter, okay? But again, active transport, moving against the concentration gradient, and then I'm using energy. Vesicular transport, we're going to get a couple more terms here. So transports large particles across the membrane. They're going to be transported inside bubble-like sacs called vesicles, which is how it gets its name. We're moving substances into, out of, and around the cell. We have two types. We have endocytosis, brings things into the cell, endo, inside. Exocytosis is going to move things out of the cell. And again, very important to remember that these are going to require energy from ATP to move things around. So here's just a little bit picture here. to sort of link all that stuff that we have together. But again, we want to just realize that we're using this vesicle, okay, to move things around. There are two main types of endocytosis, okay, and again, good terminology stuff for your lecture exams. You get phagocytosis, which is cell eating, and this is where the cell engulfs relatively large or solid material, okay, so phagocytosis, solid material. Okay, so bacteria, cell debris, and inanimate particles is going to form this endocytotic vesicle. Okay, it's going to fuse with lysosome, and then it's going to be digested. Okay, but important to remember, we're looking at endocytosis, and we're going to have these two main types, and we want to know which one is used for solid material, and then the other one is going to be used for liquids. Okay, so pinocytosis is cell drinking. Okay, so infolding plasma membrane surrounds a very small volume of extracellular fluid containing dissolved molecules. It's going to allow the cells to sample extracellular fluid so they can bring things kind of inside. Okay, and it's going to absorb nutrients, going to be non-selective. Okay, but it's going to be part of our endocytosis and part of this use of these vesicles, okay, or our active transport. Now we get to exocytosis. We're using a vesicle again. We're transporting things, or we're ejecting them, okay, from the cell interior into the extracellular fluid. So we're expelling them from the cell. Accounts for hormone secretions, neurotransmitter release, mucus secretions, waste ejecting, kind of a lot of things that we're kind of making within the cell. They're going to be closed in this secretory vesicle. It's going to migrate to the plasma membrane, okay, and then it's going to rupture and spill its contents out of the cell. So again, we want endocytosis, bringing things in, exocytosis, moving things out. They're both using vesicles, and then we want to just know, what are those main terms? We want to make sure we know phagocytosis is solid cell material. Pinocytosis is a part of endocytosis, okay, but it's for liquids. And then we want to know exocytosis, again, is just moving things, but again, it's going to account for all the things that we're sort of making inside the cell too, okay? And here's just a little picture of a real cell kind of expelling its contents into that extracellular environment, which is really, really cool. And then now we're going to take our break here, and then we're going to come back for part 2 of chapter 3, where we just look at what's inside the cell and what are the functions of all the things that are inside the cell.